Hi all, I am Simone Tiberi. I am a postdoc at the University of Zurich, where I work with Mark Robinson in developing statistical methods for analysis in bioinformatics. Today, I'm going to briefly describe a distinct, which is a collaboration with Helena Crowell, Pandelisa Matsidis, and Lucas Weber. Distinct is a very general method to detect differences between groups of distributions. And although it's very general, we mostly think of it in the context of single-cell data, where you naturally have a hierarchical structure with multiple samples of biological replicates and several single-cell measurements that are associated to each sample. In this context, this thing can be applied to single-cell RNA-seq or single-cell flow and mass cytometry data to perform differential state analysis, therefore to identify genes or markers that vary in specific populations of cells, such as cell types. Now, canonical differential methods target changes in the overall mean. But in single-cell data, you can actually have more varied scenarios, such as those ones in figure 1, and also have differential patterns where the overall mean is unchanged, such as dB here. Now, these patterns can arise, for instance, when you stimulate cells, and some cells are upregulated where others are downregulated as a consequence, but they leave the overall mean roughly unchanged. Now, these things can detect differential patterns regardless of whether their mean is unchanged or not. This is available as a bioconductor package, and we will have in 1 or 2 months a preprint with the method details and the full benchmarking. Now, the key idea of distinct is to actually look at the cumulative density function. So if you take these two examples here, so a canonical shift in the entire distribution on top, and a dB change, so differential pattern with the same mean on the bottom. If you plot the CDFs of both, you will see a clear difference between the red and the blue group in both cases. Obviously, on top, there will be a shift of the entire CDF, while at the bottom, you will have overlapping CDFs at the middle due to the fact that the mean is unchanged. But there will be differences in other areas of the CDF, particularly on the tails here. Now, this thing builds a fine grid and looks at the difference between the CDFs in the two groups at every cutoff of this grid. And by doing that, is able to identify differential patterns even when there is a difference between groups that only affects one area of the CDF or one area of the density analogously. Now, to perform a test, we then consider a non-parametric hierarchical permutation test, where instead of permuting the samples, which are typically not enough from biological data to obtain a permutation test, we permute the cells. Therefore, this thing really builds around these two key ideas. On one hand, looking at the CDF to observe differences, and on the other hand, performing a permutation test by permuting cells instead of samples. We performed several benchmarks on both simulated and experimental single-cell and flow and mass cytometry data. But here I'm only going to show you the most comprehensive simulation study, which is a simulation study from our single-cell RNA-seq data, which were simulated from Muscat, which is a bioconductor package that allows you to simulate differential patterns from each one of these 4 scenarios. We then compare distinct to some pseudo-bulk approaches, which are just bulk approaches applied to aggregated pseudo-bulk counts, because they perform the best in these preprint from Helena Crowell. Now you can see, yes, you can see distinct in red or dark red, whether we're inputting log CPMs or CPMs. And you can see that distinct has roughly the same performance as the pseudo-bulk methods on these 3 differential patterns that are here that imply a change in the mean, even though it has a higher true positive rate, and that's because the pseudo-bulk methods are fairly conservative. But the ranking, if you look at the actual ranking, it appears as to be fairly similar between methods. But it leads to a major advantage when it comes to a pattern such as dB, which is this one here that leaves the overall mean unchanged. And it's trivial because obviously pseudo-bulk methods are targeted to detect changes in the mean. And this is exactly what we aim to obtain with the method, something that would capture changes between groups of distributions or groups of single cell data, regardless of whether these changes concern the mean or not. So to briefly wrap up, we developed Distinct, which is a differential method to compare groups of distributions that does not rely on asymptotic theory, which can be inaccurate in small sample sizes. It avoids parametric assumptions that can be challenging to meet on single cell data, where you have a high degree of biological and technical variability. It explicitly models biological replicates. It's also able to identify differences that do not involve the mean and can be applied to several types of data because it's fully non-parametric and it has no assumptions of how the data were generated. And in this context, as I said, we mostly think of single-cell RNA-seq and single-cell cytometry data. It can also adjust for covariates such as batch effects and allows to compare more than 2 groups. The method is available as a bioconductor package, and a paper with details will follow, I believe, at the end of August. If you're interested or have any questions, feel free to also email me. There is my email here at the bottom of the poster. So thank you for listening.